Hey Mustangs, in this video we're going to be taking a look at the structure and function of the cell membrane. Now, if you remember way back to the beginning of the school year, one of the first things we talked about that we discussed was the importance of the structure of a molecule or an organelle, structure in general, how its structure can affect the function of something. So structure and function are really closely tied together. And the cell membrane is no exception. In fact, its very unique structure is what allows it to control what goes in and out of the cell. And so once again, we see here, as with almost everything that we see when we talk about biology, structure and function are very closely tied together. So when we take a look at the overall image of the cell membrane here, we can see that it's actually made out of 3 of the 4 macromolecules we learned about. We learned about proteins, lipids, carbohydrates, and nucleic acids. And when you look at a cell membrane, you're able to find lipids, proteins, and carbohydrates, but not the nucleic acids. So if we take a look here, we see these circles with the tails are the lipids. These purple structures are the proteins. And then these green structures that you see here, 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 all throughout, those are the carbohydrates. So proteins, lipids, and carbohydrates are the main macromolecules that are used to build the cell membrane. Also, if we take a close look here, we see that not only do we have 1 layer of lipids, but we actually have 2 layers of lipids that we call a lipid bilayer. So there's a layer of lipids here, and then there's a layer of lipids here. So we have a lipid bilayer that makes up the cell membrane. Here's another image that we can see the cell membrane. So once again, we see here the circles with the tails coming down. Those are the lipids. The proteins are once again in purple here, these structures that span across the cell membrane. Some span the entire way across, others are on one side or the other. And then you have the carbohydrates, so that's these yellowish, orangish structures that you see here. Also what you see in this image and the previous image is cholesterol. Cholesterol is a major component of the cell membrane as well. All right. One of the most important parts of the cell membrane is the lipids. The lipids make up the majority of the cell membrane, so they're really, really important, and it is the actual structure of the lipid that determines the ability of the cell membrane to control what goes in and out of the cell. So if you take a look at a lipid, the lipids that make up the cell membrane are called phospholipids, and what you see here is there is a one end of the lipid where there's a phosphate group, and that causes this end here to be attracted to water. Okay? If you remember, anything that's attracted to water we can call polar, or we can call hydrophilic. Okay? So hydrophilic means that it is attracted to water. Not only is it attracted to water though, but it's also attracted to other molecules that are hydrophilic as well. So we see that when we see a phospholipid. And then the fatty acid tails that are a part of the phospholipids that we see coming down here, those repel water. And things that repel water we call nonpolar, or tend to be nonpolar, or hydrophobic. Okay. So this end of the phospholipid is hydrophilic, and it wants to be close to water and other molecules that are also hydrophilic, whereas the fatty acid tails that come down are hydrophobic, and they don't want to be near water or any other molecule that's polar or hydrophilic. Okay, so we say this end is water loving, this end is water hating or doesn't like water, hydrophobic. Now, if we take a look at this picture here, because of this unique structure where the phospholipid has one end that is hydrophilic and the other end that is hydrophobic, this is what causes the lipid bilayer to form. So the lipid bilayer forms, so I'm going to go ahead and write this up here. So we have a lipid bilayer. Oops. So let me try that again. We have a lipid bilayer. And that is this structure here. So that's our lipid bilayer right there. The reason this lipid bilayer forms has to do with the hydrophilic and hydrophobic ends. So what you see is the hydrophilic ends go towards the outside here and the outside there, and the hydrophobic ends point towards the middle. So hydrophobic with more hydrophobic. And we can think of it as basically this is the outside of the cell, and this is like the inside of the cell. And when we look at this, the hydrophilic ends are towards the outside, hydrophobic towards the inside. And so you have this setup, and this is what forms the lipid bilayer because of the interactions between hydrophobic in the middle here, and then hydrophilic towards the outside. All right. Now, the fluidity and permeability of the membrane is determined by many different things. And one of those things is going to be the fatty acid tails. So in order to build the cell membrane, your cells will incorporate different types of fatty acid tails, or lipids with certain types of fatty acid tails. So one thing is unsaturated. Unsaturated fatty acid tails tend to have these kinks that spread things out more, and this causes a cell membrane that has a lot of unsaturated fatty acid tails to have more fluidity. So it spread things out more, it's not very tightly compact, and because of that it tends to be more fluid. Whereas saturated, a cell membrane that has more saturated fatty acid tails, you can see how much more compact this is. It compacts really close together because there is no kinks and stuff like that. So here you have more close compaction and less fluidity of the cell membrane. And this is important in order to maintain good fluidity, and it's going to be impacted by where many organisms live. So for example, an organism that lives in a really hot environment, heat causes things to move around more. So what you're going to see is organisms that live in very hot or warm environments have more saturated fatty acid tails that make up their cell membrane, because the movement you want the cell membrane to be more compact so it doesn't move around and open up as much. So they compact really close together if they live in a warm environment, whereas an organism that would live in a very cold environment, cold slows things down. So you don't want the cell membrane to be made out of a lot of saturated fatty acid tails in a cold environment. You want more of unsaturated fatty acid tails to spread things out to make it easier for things to go in and out, make the cell membrane more fluid in that cold environment where the lipids are going to want to stay still more. And then another thing that is going to have an effect on the cell membrane is going to be cholesterol. So cholesterol allows the cell membrane to maintain fluidity through temperature changes. So you don't have to constantly change if you live in a really hot and cold environment, change back and forth. No, cholesterol is something that is going to help moderate that and try to keep the cell membrane fluid as normal or common temperature changes are going to be happening around an organism. So cholesterol is important for maintaining fluidity in normal everyday temperature changes. So if we take a look, this is a more complex version of the lipid bilayer. So we have a lipid bilayer here, so we can see the hydrophobic tails towards the middle, the hydrophilic ends here. So we see that, and then hydrophobic in the middle. Now, because of this, this is actually what determines what can go in and out of the cell. Okay, so we call this we call a cell membrane semi-permeable, or another term you could hear is selectively permeable. So semi-permeable or selectively permeable, either one, it means that the cell membrane is going to be able to let some things through and other things not. So if we take a look, things that are hydrophobic, like lipids and stuff like that, can easily go through the cell membrane because of this intersection here. So the hydrophobic tails is really what's going to determine what can go through. These hydrophobic tails love other things that are hydrophobic, like lipids, but it doesn't like anything that's hydrophilic or anything that has a charge either. Anything that has a charge it's not going to want to let through. So if we take a look, large things like sugars can't go through very easily. Water, that's hydrophilic, right? It's water itself. It's not even a hydrophilic molecule that likes water. It is water. So water can't go through very easily because of this hydrophobic section. And then things that have charges like salts and stuff like that cannot pass through. So two big things to remember here is that anything that is hydrophilic or has a charge cannot go directly through the lipid bilayer. And then waste and stuff like that. There's different types of waste that the cell creates that usually aren't going to be able to go easily through the cell membrane. All right, so impermeable to polar molecules or charged molecules. So we can even add here plus charged molecules. All right, so how do those molecules get in and out of the cell? Because you need sugar to be able to go in and out of your cells. You need amino acids to go in and out of your cells. You need water to go in and out of your cells. So in order for stuff to actually go in and out of your cells, those things, and charged molecules too, are going to have to go through protein channels. So protein channels is the way, the means to the other types of molecules, the non-hydrophobic molecules, the hydrophilic and charged molecules, to go in and out of the cell. Now, if you were to actually zoom in on one of these, this is a really simplified version. Imagine like little tunnels or holes for it to go through. These protein channels here, if you were to zoom in, we see a little bit more complex version of the protein here. And you see all these little helices in the middle here. If we were to actually zoom in even more, we'd see that in the middle here, the part of the protein that is embedded in the lipid bilayer is made up of a whole bunch of hydrophobic amino acids, because you need hydrophobic in that hydrophobic section. And then towards the outside here, you're going to find more hydrophilic amino acids. So this whole thing is a protein, but you'll find hydrophobic amino acids towards the middle, hydrophilic amino acids towards the outside. All right. So taking a closer look at the different jobs or functions that a protein will do in the cell membrane. So we saw protein channels. So some proteins are there for transport. So that's what we see here. So some proteins are involved in transporting. So this is a channel protein. This is a transport protein as well. You have enzymes. So some proteins in the cell membrane are enzymes that are usually involved in cell signal pathways. So they're usually a part of a certain signal pathway in order for cell communication. Some are involved in attachment of the cell in a specific part, so that way it doesn't move around. Others are involved in cell recognition. Now this is key, remember, for your immune system. So your immune system uses proteins, usually glycoproteins, as those are the protein receptors we talked about. So when we say that your cells know what your protein receptors look like, and if it's not your protein receptors, cells of the immune system will attack any other thing that's not you, this is it right here. So glycoproteins can be involved in cell recognition. Your immune system attacks any other cell protein receptor that's not yours. So, and remember, we call those antigens. So antigens are going to be the protein receptors that are not yours. Your immune system considers any protein receptor that's not you an antigen. Other proteins will be involved in the joining of two cells. And then finally, many proteins will be involved in cell communication. Two things that we've learned is hormones. So this could be a hormone, for example, binding to a certain protein receptor on the cell. If this is hormones, this receptor would be called a target cell receptor, and it would allow the hormone to change what the cell is doing. We also saw this when we looked at the nervous system. So the nervous system, when the neurotransmitters float across the synapse and they bind to the ligand-gated channels, well, there's a receptor on those ligand-gated channels that the neurotransmitter can bind to and then sends a signal through the cell. So that's all the structure and function of the main things. So big ideas are the lipids forming the lipid bilayer, and the hydrophobic tails are going to control what goes in and out. Let hydrophobic things through pretty easily, but not hydrophilic or charged molecules. And then you have the functions of the proteins that we just saw here.